Hi students, coming to the next topic is the Eugenoids. In the last class we discussed about there is a dinoflagellates, right? Before to that one we discussed about the chrysophytes. And coming to today class, this is the third subtype which in the protestant organisms which are called as Eugenoids, and most of us we know about the Euglena, right? Euglena is a eukaryotic unicellular eukaryotic organism, right? And see, most of these eukaryotes are grown in fresh water, right? Stagnant water and damp soils. And the important point is here, most of the cases means, see, sir, you are asking Eugenoids are grown in fresh water, stagnant water and damp soils. So in these three, in which one? Okay, in which one the Eugenoids are grown? Okay, mostly. Eugenoids are grown mostly. See, the answer is here, stagnant water. Stagnant water is majorly supporting for the growth of Eugenoids. So within the stagnant water, Eugenoids are very rapidly growing, right? And the body, whatever that Eugenoid body is there, this is the Eugenoid body structure. Okay, body shows the spindle shape. The Eugenoid body having the shape is the spindle shape, right? And within these organisms, the cell wall is absent. Within these organisms, cell wall is absent. We know within chrysophytes, within dinoflagellates, cell wall is there. But in these organisms, cell wall is absent. And remember, cell wall is absent, but instead of cell wall, there is a protein-rich layer is present. There is a protein-rich layer is present. That protein-rich layer is called as pellicle. That protein-rich layer is called as pellicle. So, in these organisms, the cell wall is replaced by protein-rich layer called pellicle. Protein-rich layer called pellicle. And the presence of the protein-rich layer or presence of pellicle, these organisms shows the flexibility. These organisms shows the flexibility. See, very simple explanation. If the cell walls are there, the organisms are rigid. Body not shows any movement. Dinoflagellates, and we discussed about the chrysophytes, right? Silica made up of cell walls, very rigid structures. Body not shows any movement, but here cell wall is absent. Body shows the flexibility. Body shows the flexibility. And myonemas are present within this structure. This is one of the Eugena. Okay, there is a myonemas are there. Yeah, these are called myonemas, right? The black color structures are called as myonemas. These are myonema. These are one type of proteins which are responsible for the flexibility of the Eugenoids, or flexibility, or bending of the Eugenoid. So the bending moment or flexibility to Eugenoids given by the myonema proteins, right? And these are the organisms. First, nucleolus contain organisms. See, these are the organisms. First, nucleolus contain organisms. First, nucleolus contain organisms. Right? That is about the first nucleolus contain organisms. And in these organisms, there is an endoplasmic reticulum is there, and Golgi complex is also present. Endoplasmic reticulum and Golgi complex also present. Here there is a lysosome formation is also there. So this is about that single nucleus, and there is an endoplasmic reticulum, Golgi complex, followed by the lysosome production. That is nothing but endomembrane system. These three are considered as endomembrane. These three. Okay, first one, second one, third. These three are considered as endomembrane system. Okay? And having chlorophyll pigments. See, these organisms are having chlorophyll pigments. These are the chlorophyll pigments. Remember, the chlorophyll pigment is matched with higher plants contain pigments. Whatever the higher plants contains, chlorophyll pigments are there, and whatever the euglena contain chlorophyll A pigment is there, see these are matched. So euglena contain chlorophyll pigments are matched with higher plant pigments, right? And say below there is a contractile vacuoles are there. These are the contractile vacuoles. The contractile vacuoles are involved in the excretion process. The contractile vacuoles are involved in the excretion process. Whatever the waste products are produced within the euglena, right? All these waste products are eliminated by the contractile vacuoles, right? And reproduction is done by longitudinal binary fission. See, the organism divided from here to here. The organism is divided from here to here. So that one is longitudinal binary fission. Longitudinal binary fission. And I like to discuss about major parts within the euglena. See, if we discuss about the very starting point, see this is one of the euglenoid structure. There is a two flagella are present within the euglena. Two flagella are present. One is the short flagella, other one is the long flagella. One is the short flagella, other one is the long flagella. See, the flagellas are not equal. The flagellas are unequal. See, at that near to that flagellas, there is a small opening is there. Small opening is there. This opening part is called as cytostome. The opening part is called as cytostome. See, the cytostome is also called cell mouth. Cytostome is also called cell mouth. See, this one is supporting for the intake of food material. The cell mouth is supporting for intake of food particles. Intake of food particles. Right? Once the food particles are taken by the euglena, right? There is a cytopharynx. This is again small opening is present here. This is cytopharynx. The structure is called as cytopharynx. This one is also called cytopharynx or gullet. Cytopharynx or gullet. See, this part is called as cytopharynx or the gullet. This part is called cytopharynx or gullet. See, this one is responsible for the phagocytosis. This one is responsible for the phagocytosis. So, cytopharynx, this one is also called gullet. In this region, the food digestion takes place by the phagocytosis process. Intake of solid food material, the food is digested, right? See, that one is called as phagocytosis process. The food is breaking down into the very small pieces, right? So this is the phagocytosis process. Again, whatever the excess amount of food is there, the excess amount of food is stored within the reservoir. Within the reservoir. This is the reservoir region. So within the reservoir region, whatever that cell contain excess amount of food is there, the food is stored. There is nothing but storage food. Storage food. See, storage food in the form of paramyelin. Within the euglena, the storage food form or the storage food is called as paramyelin. Right? That is about that. I discussed here major 3 points. One is that cytostome, other one is that cytopharynx and reservoir. These 3 are related to that intake of food, digestion of food, storage of food. Right? And we discussed already there is a short and long flagella circle. And next one, sensory stigma. There is a drawn here the red color. This is nothing but sensory stigma. Sensory stigma. See, the particular region have the sense with the light. The particular region have the sense with the light. Means, for suppose euglena performs the photosynthesis process because here chlorophyll A pigments are there. This is nothing but photoautotrophic organism, right? But here the light identification process, the availability of light is done, or the availability of light is identified by the sensory stigma. And this one is also called eye spot. This one is also called eye spot. Remember, the sensory stigma, in which region the light is available, the light is available is identified, and by the help of flagellas, the euglena moving towards the light direction. By the help of flagella, the euglena moving towards the light direction. Right? that is about the sensory stigma and eye spot. Remember, the sensory stigma or the eye spot majorly having the cherubinoid pigments. Majorly having the cherubinoid pigments. Cherubinoid pigments are present, right? So that is about that sensory stigma and eye spot. So here short flagella, long flagella and reservoir storage of food material. So this is about the upper portion. And next one. See, we already discussed nucleus is there and first nucleus contain organisms, endoplasmic reticulum, Golgi complex, and these are the lysosomes. These are the lysosomes, right? And see here there is an parallel arrangement is there. The longitudinal arrangement is there, right? See, these are nothing but myelema. These organisms are myelema, and there is a chlorophyll A pigments are there, right? Chlorophyll A pigments are involved in photosynthesis, and contractile vacuoles are involved in the excretion process. Contractile vacuoles are involved in the excretion process. And remember, there is a cell wall is absent here. The cell wall is replaced by the rich protein layer called epithelium. So, presence of pellicle, the organism shows the flexibility. The organism shows the flexibility, right? That is about the euglena structure. And next one, we will discuss about the nutrition. We know that mixotropic nutrition is exhibited by the eugenoids, right? And the last point is the pharmula stage. We will discuss about these 2 points. Coming to the next topic is the nutrition process. Whatever the euglena organisms are there, these are very strange organisms because we know here when the light is available, that time these are act like in photoautotrophs. When the light is available, that time these are act like in photoautotrophs. But when the light is not available, that time these are act as heterotrophs. That time these are act as heterotrophs. See, light availability and chlorophyll A pigment, photoautotrophic nutrition. These are act like photoautotrophic, photoautotrophs, nutrition is the photoautotrophic nutrition. When the light is absent, these are grown on decaying parts. Mostly these are like sarcophags. Mostly these are like sarcophags. When the light is not available that time. So a single organism can exhibit both autotrophic and heterotrophic nutrition. That nutrition is called as mixotropic nutrition. That nutrition is called as mixotropic nutrition, right? And pharmula stage. Pharmula stage means whatever the euglena is there, that euglena during unfavorable conditions losses the 2 flagellas forms the round shape. Forms the round shape. So that round shape, this round shape indicates here. This round shape, okay, indicates here. For suppose any euglena forms the round shape. Okay, that one is indicates the euglena presented unfavorable conditions. The euglena presented the unfavorable conditions. See, if the body existed like this during unfavorable conditions also, that time its losses the water, the euglena will be there. So this is one of the protection mechanism. When its losses the flagella, when it forms the round shape, and it have the resistance to unfavorable conditions. Unfavorable conditions, maybe the high temperature, okay, and dry conditions. That is nothing but desiccating conditions. So this organism have the resistance to unfavorable conditions. This stage is called as pharmula stage. This stage is called as pharmula stage. And I like to discuss one important point. In the last class we discussed about the red tide. And there is a red sea. Red sea is different from red tides. Red sea is different from red tides. See, the red tides caused by the dinoflagellates. And especially gonolix organism. And red sea is caused by bacteria, especially cyanobacteria. Right? Bacteria is Trichodesmium, Trichodesmium, Erythrium, Trichodesmium, Erythrium. Right? This is a bacteria. This is a bacteria. So Red Sea caused by the cyanobacteria. Organism is Trichodesmium, Erythrium. See, the red plates caused by the dinoflagellates. Right? The organism name is Gonoids. Right? That is about. This is one of the important points. Right? So in the next week we will discuss about the next topic.